All right. Hello, and I'm really pleased to be here to share some of my work. This may be a little bit of a different flavor. I'm going to talk a little bit about biology and the approach that I've taken for integrating biological and Earth surface process models. So I'll start with this map of species diversity across Earth's continents. And there's a few important patterns to note. There's more species located near the equator. And there's also, of interest to us as geomorphologists, a lot of species distributed in mountain ranges. And I've highlighted some of these places that kind of pop out on this map here. This shows that the residual of a correlation between climate variables and species richness, again, across the world. And again, you can see that some of these mountain ranges, like the Andes, the Himalaya, Central African Rift Valley, are some notable mountain ranges that kind of pop out as red. So local climate alone cannot explain the high species richness of mountain ranges. And this suggests that there might be a link between geologic processes, mountain building, topographic evolution, and the diversification of life, which is exciting. So how would this work? So today and in my work, I'm generally focusing on what's called allopatric speciation. And so a phase of allopatric speciation is thought to accompany or initiate at least most speciation events. So let's say we have some bunnies on a landscape, and they can migrate across this flat landscape. A mountain range grows, and they find it more challenging to migrate across this landscape. They accumulate genetic differences, and then we can resolve the relative ordering of how lineages may become separated from each other, and perhaps we may be able to actually infer information about absolute time as well, although we may have some more work to do before we get there. So this would suggest that, okay, in places with more topographic change, we should find higher species diversity. So this is a map of mammal diversity across North America, and there's generally more mammal species in the western half of the continent, where you have more topographic complexity. But for freshwater fishes, this is not true. You have your highest species diversity on the eastern or tectonically inactive margin. And this is true not just for fishes, but also crayfishes, salamanders, mussels. The southeastern US is an exceptional biodiversity hotspot when compared to other temperate regions across the world. I wanted to understand this on a global scale, so I categorized the world's mountains as tectonically active or inactive. And if you don't like my categorizations, I'm open to feedback. Then I counted the number of species per unit area in these different geologic settings. And I'm showing a histogram of these results for mammals on the left and freshwater fishes on the right. And so the point is that there's more species per unit area for mammals in tectonically active mountain ranges as compared to tectonically inactive mountain ranges, but that difference collapses for freshwater fishes. I also repeated this analysis for squamates, so these include reptiles like lizards, snakes, turtles, crocodiles, et cetera. And for terrestrial taxa, they are more diverse in tectonically active mountain ranges, but this is not true for the freshwater taxa. So are tectonically active mountain ranges more biodiverse? Perhaps yes for terrestrial taxa, but no for these freshwater taxa. And this motivates my work. When we think about, I drew a polygon for this landscape where the bunnies lived, but if we think about the biogeographic arenas for freshwater fishes, these are river networks. And we know that river networks can change their connections to each other over time, and that this can split and merge lineages of freshwater organisms and may present opportunities to initiate allopatric speciation. So let's say we have a river flowing across this sort of plateau. We have a neighboring river across the drainage divide flowing down an escarpment type situation. And there's a fish that lives on this plateau side. Say there's a river capture. So the headwaters of this plateau draining river get rerouted into this escarpment draining river. And this fish population may find itself in the wrong place on the opposite side of a drainage divide, and it can accumulate genetic differences. And then again, we can infer the pace or geographic arrangements of these, the history of this escarpment and these river captures using molecular data and things like phylogenetic inferences derived from molecular data sets. And something that's exciting, I think, is that we can interrogate the phylogenetic record at different timescales. And today I'll mostly be talking about the relatively recent timescale, but I'm excited to think about how we can detect things in deeper time that may not be immediately apparent in topographic data sets. So when I started to work on this, I was not working with an evolutionary biologist, and so we decided that we should approach this using some simulations. So I'm going to walk through those simulations, and then I'll move on to some sort of ongoing work in my research group at Florida State that is exploring these things in an empirical setting. So we coupled a landscape evolution model with what's called a meta-community model, and I'll talk more about that in a second. But basically, this is a stream power incision model. And to induce river network reorganization into this model, we introduced a dipping fault. So this is just a plane of more erodible rock, and it's dipping towards the lower right-hand corner of the domain. So as the surface of the landscape lowers, it intersects with this fault, and the fault trace migrates across the landscape. Then we seeded this model with, this is like an agent-based model. And so we seeded it with these organisms, and these organisms can be born, die, move throughout the river network, and then we also have speciation that occurs. So I'll just play the landscape evolution model for you for fun, perhaps. Spacebar. Okay, thank you. Okay, so you can see that as the rivers are eroding into this plane of more erodible rock, they expand, and then these little blobs are some small river captures that happen. This is a typical time series from one of these simulations, and so this is model time on the x-axis, and then the relative size of these river captures. So we have these little pulses of river captures throughout the simulation. This is showing the speciation rate of the average speciation rate of the organisms throughout this model. And I'm comparing a dynamic model, that's the black line here. And this is basically the coupled model, so where we have the changing landscape and we have the meta-community model running on top of that. The gray line is a static landscape, so we just have the meta-community model running on a landscape that isn't changing. So we get these bursts of speciation associated with these river captures. But we also see these little pulses of extinction, and that's because these basins shrink out of existence, and it causes some of these lineages to go extinct. So we can difference these two and get a diversification rate. And on the whole, we have an elevated diversification rate despite these little blips below zero as these basins shrink out of existence. But the net impact of these river captures depends on both the size and the frequency of these river captures. So if we have bigger captures, we have more we have a bigger biological impact, as well as rates of molecular evolution and the dispersal capacity of the organisms. And this model produced similar results to that of Lyons et al. 2020 species evolver, which was incorporated into the land lab framework. So if we can inform on the landscapes in which there are frequent river captures, how big these are, maybe we can start to better constrain the impact that this may have on species and explain some of these patterns of biodiversity. So with that, I'll move on to an empirical setting. So I've been working here for a while. This is the Eastern Continental Divide. It separates rivers that flow into the Atlantic Ocean from those that flow into the Gulf. And it is in motion, and particularly along the Blue Ridge Escarpment, which goes from Georgia up to Virginia. There is asymmetric topographic relief and slopes across this drainage divide. You can see this escarpment here, which is a steep landform on the Atlantic side of the escarpment, and people have been describing this in the literature for a long time. Davis described the contest for water across this drainage divide as the most stubborn contest ever waged. We can apply different topographic proxies for inferring drainage divide instability. This is a Chi map, which indicates, again, that this drainage divide is moving towards the northwest. So we wanted to test these different topographic proxies, Chi or Gilbert proxies, et cetera. And so I measured paired Brilliamton-derived erosion rates from catchments across the drainage divide. And I won't go into too much of the details, but here I'm showing a delta chi versus an inferred horizontal migration rate. And then this is a difference in channel head slope. And anyway, they both generally suggest the correct relative direction of divide migration. But another take home is that the rate of horizontal divide migration is very slow in this landscape. So like, tens of meters per million years, and perhaps this is how we maintain these passive margin escarpments for a very long time. But we also have evidence for river captures. So that diagram was inspired by this setting. So we have nick points in capturing rivers, we have barbed tributaries, we have sedimentary deposits with provenance that suggests an origin from outside modern catchments, and we have wind gaps. So we might expect that there should be slower erosion rates in tributaries upstream of the nick zones and higher erosion rates within the nick zone. So we again collected Brilliamton-derived erosion rates to test this. So this is the Dan River. It flows to the Atlantic, and you see it's taken this huge bite out of the escarpment. There's this somewhat impressive for the East Coast gorge here or nick zone. And the idea is that this river used to flow sort of parallel to the escarpment and go into the Ohio River system. So we do see that there's about a 3 times difference in erosion rate for a tributary within the nick zone as compared to upstream of the nick zone. So I wanted to see if we could try and date this river capture using a 1D stream power incision model and try and match it basically to our observations of the position of this nick point as well as the measured erosion rates. So this is how the model is set up. There's some initial condition and there's uncertainty about where this river capture point should be. But so the upper part here, we basically allow that to flow into this Paleo Upper Dan River at the start of the simulation. And then the nick point migrates upstream. And we also track the erosion rate in this basin this catchment where we measured this elevated erosion rate. And I went through a whole sort of, explored a wide parameter space and did a model misfit sort of exercise and tried to minimize the misfit. And this is showing the frequency of local minima in this exercise versus the age of the river capture. And the answer is rather messy, but it seems like either the capture happened relatively recently, we have a peak here, or perhaps it was older, around 7 million years ago. And a lot of this uncertainty has to do with where the capture position happened, as well as we varied the value of the slope exponent n. And that n is why there's a bimodal distribution for the answer. So let me move on to the biology. So in addition to these nick points, et cetera, we have zoogeographic evidence for river rearrangements across the Blue Ridge Escarpment. So there are some species that are widespread west of the drainage divide. So here I'm showing the Tennessee River in purple. So there are many species that are widespread in the Tennessee River, which drains into the Ohio, and then they have these sort of odd disjunct distributions across the drainage divide. My student Alexis Coley is working on one of these species. This is called the Saffronshine or Hydrophlox rubicrocius, and it is distributed again in the Tennessee River with these few disjunct distributions and headwaters on the Atlantic coast. And these places where it's found have these nick points that are consistent with the history of river capture, including in the Savannah River and the Linville River. So we use tissues collected from across this range. We sequence the DNA from these individuals, and I'll show you some of these results now. So this is showing a phylogeny. Each of the tips on this tree represent an individual, and then the lines show how they're related to each other. And I'll call out the things that you should pay attention to. So the Linville, which is on the Atlantic coast, is sister to the North Toe, which is on the western side of the drainage divide. We see the same relationship for the French Broad, western side of the drainage divide, and Savannah, eastern side of the drainage divide. This is a sort of clustering analysis. It's comparable to 23andMe. So each of the bars is an individual, and we determine an optimal number of clusters and then assign figure out the proportion of ancestry that would explain the observations. And so the French Broad and Savannah come out as two separate clusters, which suggests there's a significant amount of genetic differentiation between these two rivers. The Linville and North Toe come out as one cluster, which indicates there's comparably less genetic differentiation between these two rivers. But something that we were surprised by was this admixture. So you can see there's yellow bars in the Linville between the Linville and the Watauga River, which we weren't quite expecting, and I'll dig into that in a minute. So let's move on to the Linville River. So here is a topographic map of the Linville River Gorge. The Linville River flows to the Atlantic coast, and again, it has taken this big bite out of the plateau side of this environment. It's a really cool place. It's one of the few wilderness areas east of the Mississippi River in the US. There's old growth forest in the gorge because it was too steep to log. And there's nice waterfalls, hiking trails, etc. So I recommend it as a visit. Brad Johnson published a paper in 2020 kind of describing an idea for the relative chronology of river rearrangement in the Linville Gorge. So the North Toe, Nolichucky, this flows into the Ohio River system, flows through this valley here. And so the idea is that this Linville used to flow into the North Toe, Nolichucky, and then was captured by this Atlantic Slope River. And again, you have this nick point in the river that supports this. Recall that the Linville and the North Toe populations of the saffron shiner are sister to each other, which supports this sort of T-bone rearrangement between these river systems. But we saw this admixture between the Linville and the Watauga, which was unexpected until we zoomed into the DEM. And in the headwaters of the Linville River, here's the Watauga, which again, this flows to the Ohio, Linville flows to the Atlantic. You can see it's a very unimpressive drainage divide. It's rather flat. There's in fact a swamp right here. And it looks like maybe there's some sort of wind gap migration occurring. In this analysis, you can, instead of assuming that evolution must occur through these bifurcation events, you can allow for migration between different lineages. And so Lexi's analysis resolved this migration event between the Watauga and the Linville, which is notable because the Watauga is rather distantly related from this Linville lineage. So Linville and North Toe remain sister to each other, but we have this sort of second migration event from another population. And I'm excited to think about whether this genetic data and analyses can inform on when this migration event occurred. And I'll note that these lineages are too recently diverged to accurately use molecular clocks. You may have seen the use of molecular clocks where you assume some rate of molecular evolution to date things in absolute time, but this isn't appropriate for the study system. So I want to introduce you to a rather new simulation approach that we're using. And so we're trying to employ this simulation framework called SLIM, which I took this from the manual, but they originally we're calling this selection on linked mutations. They're studying a specific evolutionary process, but now they want to call it simulating life in machines, which I think is I thought might appeal to this audience. And basically, this is a flexible open source framework for population genetic simulations. And it's a really impressive modeling system. I won't go into the details. You can do all sorts of things, including spatially resolved models, but basically individual organisms and their genetic information is resolved. So it's appropriate for comparing to these population genetic analyses. And so these results are rather preliminary, but I wanted to explore what the population genetic differentiation would look like in a scenario where we basically have repeated river captures. So the Blue Ridge Escarpment has been continuously migrating inland for hundreds of millions of years. And so you have this repeated movement of organisms over the drainage divide. What does that do? So I set up a really sort of simple framework where I have a parent population that sends a daughter lineage over the drainage divide. It becomes genetically different. And then at a later time, we have another lineage, another river capture, and a new set of organisms gets moved across the drainage divide, and then you have bidirectional migration between these daughter lineages. So in these preliminary results, what I'm showing on the y-axis is called FST, which is a metric of genetic differentiation. And here I have a scenario where there's just one river capture, 2 river captures, and 3 river captures. And then I vary the size of these river captures. And the point is that as you have more river captures, you lower the degree of genetic differentiation across the drainage divide. And anyway, so I'm still working in sort of relative space and thinking about how to calibrate this to the real world, but I'm excited about, I wanted to share it with you because this is a modeling conference and I think this is an exciting tool. So I'll wrap up. Fluvial environments and tectonically inactive landscapes like the Eastern North America remain active. Perhaps this could explain this observation that these freshwater fishes are just as diverse in tectonically inactive mountain ranges as they are in active mountain ranges. Drainage basin boundaries are dynamic, and the movement of these drainage divides can provide opportunities for allopatric speciation. And biological data, both phylogenetic information in deeper time as well as population genetic information in more contemporary time scales could inform on the frequency and age of river rearrangements. Special thanks to my research team who have helped move this research forward since I started my position at Florida State, my students Lexi, Kat, and Malia in particular. And with that, thank you, and I'm happy to take questions.
Thank you, Maya. Any questions? Jeff.
It's a good question. The question was about what the impact of range size and the relative importance of these river rearrangements for range size. One thing that we learned from the simulations I showed early in the talk, so basically when we reduce the dispersal capacity of the organisms, we get small range sizes. And something that happened is that they went extinct all the time. And so I hear what you're saying. You may impact more things, right, if you have a larger range size, but I think you do run into the risk of extinction as well, and you might have to think about both processes. So yeah, good question.
Do you see the species to help lower the uncertainty for the model species? I wonder if you choose a species also based on the time that it takes for genetic changes to occur based on that, or whether you select those species.
Yeah, so the question was about whether we could use different species and leverage differences in, for example, rates of molecular evolution or something to sort of learn about different components of the landscape. So Lexi is working with a few different species. I think that the rate of molecular evolution is probably similar across them, but something that is different is their range sizes, their population sizes, and then their life history. So she's working with minnows and also darters. So minnows have large populations, they live in the water column, and darters are benthic and have smaller population sizes. And so that's one thing we're considering. I think also we're trying to start a crayfish project as well, because those are aquatic but can also move over land. So we may see some interesting differences there. But these crayfishes also have similar disjunct distributions across the drainage divide. So